eyes of the scientist look at the world of nature and seek to understand it. Marcello Malpighi of Bologna was one of the first to use lenses to extend the range of the scientist's vision. These first microscopes could survey the world and its creatures in detail undreamed of. In 1661, Malpighi made history by observing the movement of blood in the fine veins of a frog's tongue. In the early days of the Royal Society in London, Robert Hooke designed other microscopes and examined the structure of a host of things and creatures. He too astonished his world by his detailed enlargement of Pulex irritans, the human flea. At the same period, the mid-17th century, a Dutchman called Van Leeuwenhoek began to grind minute single lenses of unexampled power. Seeking the cause of the hotness of pepper, he left some grains of the stuff in water. 3 weeks later, he found growing in the water incredibly many very little animalcules. The smallest of these we now call bacteria. These were the beginnings of the light microscope. Over the centuries, its aberrations and distortions were corrected. Magnifications of over 1,000 times became possible. By 1900, the optics of the light microscope were substantially what they are today. The light microscope is still essential to the scientist. Yet there is a limit to the detail it will reveal. Some objects are so small that they cannot be clearly seen. Others are too small to be seen at all. And this is due to the properties of light itself. The behavior of light waves can be simulated in water with a ripple tank. Light from a given source has a measurable wavelength. An object whose width is greater than half the wavelength of light distorts the pattern of the wave movement, and so the eye can see it. But a smaller object hardly deflects the wave movement. This means it cannot be seen. To see and magnify such small objects, we must find an alternative to light with a shorter wavelength. With light, glass lenses are used to focus the beam. A stream of electrons in a cathode ray tube has a wavelength thousands of times shorter than that of light, and it can be focused with an electromagnetic coil. As the current in the coil varies, the beam is brought in and out of focus. Thus, an electromagnet provides a new kind of lens for focusing a beam of electrons traveling along its axis. It makes possible an electron microscope. What is needed? First, an electron gun to produce a stream of electrons. An electromagnetic lens to act as a condenser, focusing the stream onto the object to be examined. The electrons pass through the object as light passes through a slide. Then another magnetic lens, the objective, to create an intermediate image. Finally, a magnetic projector lens to throw a further enlarged image onto a fluorescent screen or a photographic plate. The electron gun must work in a vacuum. So the column must be solid and vacuum tight, with the fluorescent screen viewed through a window. Here is a modern electron microscope. The cable at the top is connected to a high-voltage electrical supply, up to 100,000 volts. Inside the casing is the electron gun. The electron beam is emitted by a heated V-shaped filament mounted in the cathode. Electrons are attracted to the highly polished anode. They pass through the hole in the anode plate and on down the column. Below the anode plate is the condenser lens, which focuses the electrons onto the specimen. All the lenses are simply electromagnetic coils in metal cases. Below the condenser lens is the specimen chamber, with its window and door. In the chamber is an airlock which can be pumped out. The specimen itself is put in the little copper holder mounted on a grid. The grid is so small that it is difficult to see the grid squares without a magnifying glass. Below the specimen stage is the objective lens. The image is focused by varying the current in this lens. There are two projector lenses at the bottom of the column. Below them is the viewing chamber. The image is projected onto the viewing screen, which can be raised to expose a photographic plate. The resolution of photographic emulsion is normally at least 6 times that of the human eye, so that an enlarged photograph reveals more detail than can be seen on the viewing screen. The electrons cannot penetrate more than 1 millimeter in air, but in the microscope they must travel from the electron gun to the viewing screen. They can cover this distance only in a very high vacuum. The world of the electron microscopist seems totally unfamiliar. An abstracted world of shapes and planes which at first mean nothing. Yet this is the detail of the real world. This is the ultrastructure of real things, of a chemical smoke, of soap, of chalk, a human tissue cell, etched steel, and at the extreme power of the microscope, the atomic structure of a platinum compound, parallel lines of atoms 1/1,000,000th of a millimeter apart. If a heavy current is passed through 2 carbon electrodes in a vacuum, carbon will condense on a slide beneath. The carbon forms a sheet 20/1,000,000ths of a millimeter thick, which can be floated off. It is thin enough to be easily penetrated by a beam of electrons, so it can act as a support for specimens many times smaller than the holes in the grid. A suspension of latex rubber is being examined. The rubber is supported by the carbon layer. Preparing specimens for the electron microscope is a painstaking job because everything is so minute. The next step is to put the holder into the specimen chamber, close the airlock, and pump it out. At the lowest magnification, the squares of the specimen grid are visible. The operator selects one square. Already the latex particles can be seen sitting on the layer of carbon. At 20,000 times, magnification is changed to the higher range. We can see that spongy latex rubber is actually built up of very many small spherical particles, each about 1/1000th of a millimeter in diameter. Magnification on the microscope screen, 36,000. On a 10-foot wide cinema screen, 1.5 million. By taking a picture with the built-in camera and enlarging the photograph, magnifications of 2 million times or more are possible. The prints are known as electron micrographs. A freshly broken piece of plate glass is one of the sharpest cutting edges known. It can be used to shave off sections of plant and animal tissue so thin that an electron beam will pass through them. The cutting instrument is called an ultramicrotome. The slices are 10 to 30 millionths of a millimeter thick. The thickness of a slice is judged by its interference color. Normally, the tissue is embedded in a plastic support. Thin tissue sections allow the botanist or the anatomist to observe structure and ways of growth. For instance, in the sheath round the fibers of the sciatic nerve. In a 4-day-old mouse, the cell membrane appears to be folding inwards around the nerve core. As the mouse develops, this spiral loops and loops again. By 16 days, the protective covering is tightly wrapped about the nerve itself. But many specimens are too dense to be penetrated by electrons. If the specimen is put on a slide beneath the carbon rods, it is possible to make a cast or replica of the surface of the specimen. When a current is passed between the rods, the carbon vaporizes and then condenses all over the specimen, making the cast. Now the specimen itself is removed so that only the cast is left. The carbon cast can easily be penetrated by electrons. Yet an electron micrograph gives a poor impression of structure. This enlarged model of the cast shows how the problem has been solved. The cast is shadowed with a heavy metal, such as platinum or gold. With an actual specimen, this is done in a vacuum. The resulting electron micrograph is reversed to make the shadows black. Another way of throwing a surface into relief is to direct the electron beam onto the specimen at a low angle. This creates long shadows. The technique is used to study the surface of metals, as with this micrograph of a fine scratch on a sheet of copper. A new improvement to the electron microscope projects the picture onto a television tube. A single bacterial cell is being examined. The electron beam falls directly onto an electronic camera tube below the viewing screen. This image intensifier increases the brightness level, allowing higher magnification or the use of lower electron intensities. The intensifier can be used in the study of wear. A standard laboratory machine records wear between a brass rod and a steel plate. A smaller version can be made to fit inside the electron microscope. The tendency today is to put the laboratory into the microscope, as it were, so that experiments can be watched as they happen. Here Here, the brass pin on the left is set at an angle to the electron beam, casting a shadow to the right. Magnification is 10,000 times. On the normal viewing screen, the picture is hardly visible. But with the image intensifier, one can see pieces of the pin breaking off and welding themselves to the plate. In many industries today, the electron microscope plays an important part in research and production. The plastics manufacturer uses it to help analyze the structure and properties of synthetic materials. In this sample of PVC, he can check the shape of particles and the distribution of different particle sizes, which will affect the properties of the plastic. The paint makers expose samples to wear by light and water, simulating years of use in a short time. A weathered sample can be compared with an unused one. A carbon replica shows that the unused paint has a generally uniform surface. But in the weathered paint, the binding material has worn away. Many synthetic clothing fibers generate static electricity when they are worn. Before use, the surface of a thread of nylon is fairly smooth. After it has been worn and washed for a few weeks, static electricity leaves burn marks on the fiber, pitting and distorting its surface. Nylon, equally worn, but washed in an anti-static detergent, remains almost as good as new. The botanist, studying the effect of weed killers, takes different weeds and makes replicas of their leaf surfaces. The leaves of poppies and many other weeds have a surface which is porous to liquids. But the corn marigold has an elaborate structure of wax on the leaves, which makes them waterproof. This may explain why the marigold is immune to contact weed killers. The scientist studying air pollution takes samples of air from tunnels and city streets. With the electron microscope, he can distinguish the impurities which we inhale. Short chains of carbon expelled from the exhausts of vehicles, and large carbon particles from domestic chimneys. Years ago, it was predicted that a raft of bubbles could simulate the behavior of the atoms in a metal crystal. If the raft is strained, faults or dislocations run through the lines of bubbles. Now these dislocations can be seen in the microscope, in a film of aluminium. The electron beam heats and stresses the metal, causing the dislocations. Now watch a film of duralumin being cooled and heated. Duralumin is a solution of copper in aluminium. As the metal cools, dark patches appear. A copper aluminium compound is being precipitated. As the temperature rises, the precipitate redissolves. Watch a crack spreading in aluminium. It has never before been possible to see the way metals behave almost at the atomic level. The biologist, too, can confirm predictions. In the past, many diseases were attributed to minute living particles that had never been seen, the viruses. Today, many viruses can be grown in tissue cells and sprayed onto specimen grids. The scientist can take polio, for instance, and watch the way the virus replicates inside the cell. Or he can slice bacteria into sections and study different stages of attack as a bacterial virus colonizes the cell. In these smallest forms of life, the microscope reveals precision and order, as with adenovirus, parasite of the throat and chest, only 30 millionths of a millimeter in diameter. Our knowledge of it has grown until today we observe its precise structure, 252 protein units in a regular 20-sided solid. At its core, hardly discernible, is the thread of nucleic acid, the complex molecule that determines the growth and function of every form of life. Yet this microscope, which seems to bring us so near to the heart of things, is little more than 20 years old, as primitive in its own field as a 17th century instrument. But unlike Malpighi or Leeuwenhoek, the physicist understands the theoretical limitations within which he can improve lenses or step up voltages. Already, the electron microscope has shown how intricate is the detail of everything, and how much more there is to learn of the ultrastructure of our world.